Hello, everybody. Thanks again for joining this lecture. Now, today we're going to be talking about microbial growth. We're going to try to cultivate or grow microorganisms. You probably know by now that to grow anything, to cultivate anything, you're going to need food and you're going to need the proper environment. And so we're going to talk about each one of those separately and try to see how we can cultivate microorganisms. We'll be doing a lot of this in the lab also. So it's something to look forward to in the lab if we have already not done it there. Now, like I said, we need food and we need a proper environment. So what does the organism require in food? So we're going to talk a little bit about some of the inorganic requirements first, some of those elements or atoms that we will need. And most of those are found in the crust of the earth, bodies of water, and in the atmosphere. For example, we have oxygen. That's needed. It's a major component of all the macromolecules and other inorganic compounds, organic salts such as sulfates and phosphates and nitrates. And of course, water itself has oxygen in it. It plays an important role, oxygen that is, in the structural and enzymatic function of the cell. Hydrogen, a major element in all of our organic and several inorganic compounds. We talked about the water, the salts, certain naturally occurring gases like hydrogen sulfide, methane, hydrogen gas. And it is the hydrogen that helps to maintain pH. Very useful in forming hydrogen bonds between molecules. Nitrogen, without nitrogen, we're not going to have our proteins or amino acids. Most bacteria can decompose proteins to extract nitrogen from them. Others will use ammonium compounds or nitrate compounds. And a few bacteria can even fix nitrogen, a process where the organism can make or build other compounds from nitrogen. And these compounds can be used by other living organisms. Phosphorus, sulfur. Sulfur, again, you can find those in some of the amino acids, not all of them. And it's one of those common material that is used as an electron acceptor when oxygen is absent. A lot of your anaerobic organisms, you need to use not oxygen as the final electron acceptor, but they can use something like sulfur. Some bacteria can use sulfates, even hydrogen sulfide as energy sources. Phosphorus, the phosphate group, is found in DNA and RNA and ATP. We can have those, nucleic acids and ATP, without. Every nucleotide has that. You remember the three components: a phosphate group, a sugar, usually ribose, deoxyribose, and a nitrogen base. So you cannot have those compounds without a phosphorus group. And you can have them from phosphorus from other sources like hypochloric acid and things like that too. Right? Some other elements that are needed. You have this slide there. I can just read from the slide, or you can read it on your own. Usually sodium, we talk about the sodium pump in a lot of the transport in cells. Calcium is a good stabilizer in cell wall, and usually the spore coat of bacterial spores. Magnesium is a component of chlorophyll. We're not going to be talking about these photosynthetic organisms. They're generally not human pathogens, so we'll probably not be talking much about them. Iron, an important compound in cytochrome, and when we do respiration in bacteria, when we do bacterial processing, processes for energy production, we will talk about the electron transport chain. And in there, we'll talk also about some cytochrome oxidase that are produced to carry out some of those reactions in the electron transport chain. Zinc, usually a regulatory element in eukaryotic genetics. And potassium is essential for protein synthesis and some membrane function. Now, some organic requirements. Well, organic compounds, every time you see carbon, and carbon is present in something, they're usually organic compounds. So it revolves around the carbon element then. Organic nutrients contain carbon and hydrogen atoms, and usually they're the products of living things. They're found in all the macromolecules, carbohydrates, lipids, proteins, nucleic acids. Okay, and they can be extracted from other molecules like carbon dioxide, methane, citrate. We will talk about the citrate test in the lab where organisms can extract carbon from citrate and be able to grow. Now, here we have all the elements that we need that are required in the food supply or environmental supply. So we want to know now, how are these things crossing the cell membrane to get into the cell so they can be used? Well, a lot of this is probably going to be a little review for you, but the basic question in mind is, how do nutrients cross the cell membrane to get into the cell? Well, one of the ways, diffusion. Now, diffusion is usually, you all know by now, is the movement of molecules and atoms from a high concentration to a lower concentration. That, natural propensity of molecules and atoms is to be in a state of random movement. You're constantly moving. So diffusion is just that movement from a higher concentration to a lower concentration. Until the concentration is balanced, diffusion will not stop. Now, there is simple diffusion, that's what it is. There is also facilitated diffusion that can go against the concentration gradients. But here the molecules, they will bind to a specific receptor on the cell membrane and is carried to the other side. Regardless, diffusion, whether simple or facilitated, we usually do not use energy for that. Now, osmosis is usually the diffusion of water across the membrane. And usually this movement of water will continue until there is an equal amount on both sides. And we refer to that as isotonic conditions. Now, active transport is the transport, well, it may be against a diffusion gradient, or it could be in the same direction as the, but it's at a faster rate than diffusion alone. active transport usually requires some energy. Materials that are actively transported are too large to go through the pores of the membrane. And even though many of the organisms can produce hydrolytic enzymes to hydrolyze some of the large macromolecules, for example, starch, you can hydrolyze the starch to give you plain glucose molecules, but even the glucose is too large to go across. So you need that material, you need that to be actively transported, which means you are producing enzymes, permeases, and pumps to bring the material from one side to the other. All right. And most of those are like not, they're fairly large, like even the monosaccharides or the amino acids, organic acids, phosphate, metal ions. They're actually physically moved over using some enzymes and pumps. This, like I said, requires some energy. Another way to bring things into the cell is what we call endocytosis. Now here, what happens, the cell kind of encloses the material and brings it into the cell, usually into a vacuole of some sort that it creates. It's what we call engulfing the organism or the material, whatever that material is. You might have heard of phagocytosis in amoeba and some of your white cells, where in the case of your white cells will push out some pseudopods and engulf bacteria or other pathogens that are trying to invade you. And this way they can bring it in and try to pour lytic enzymes on them and digest them, or at least kill them. Okay. Amoeba will do the same thing as source of food. Maybe they see a yeast cell out there or some whole particle, and they will push out the pseudopods and bring it into what they create, a food vacuole or something, and then digest that. In the case of pinocytosis, you're doing the exact same thing as phagocytosis, but instead of solid particles, you are engulfing liquids such as oils, fats, any kind of molecule in solution. They usually refer to phagocytosis as cell eating, pinocytosis as cell drinking. But those are ways to get material into the cell, and those materials could then be used as energy sources. Now here in this slide, we're trying to group microorganisms according to their nutritional requirements. And we are showing here that there are different groups of organisms, nutritional groups. The first one we're going to say autotrophic organisms or autotrophs. And these are organisms that they have the ability to make their own food. Let's put it simply that. They could use carbon dioxide from the atmosphere and change that into compounds like starch that can be used by itself and other microorganisms for food. So these are the organisms that are never going to be nutritionally dependent on other organisms. And if they're not nutritionally dependent, they're generally not going to be pathogenic. All right? So these organisms, if we want to subgroup them, photoautotrophs, they derive their energy from the sunlight, and carbon dioxide will give them their carbon source. Chemoautotrophs, energy from inorganic compounds and carbon from carbon dioxide. Point is, though, they are not going to be dependent on you for food, and hence they will not be pathogens to you. And most of the time in this class, we're not going to focus on the pathogenic, on the non-pathogenic organisms. We are trying to learn about some of the pathogens so we can know how to recognize them and how to treat them and how to make sure we don't contaminate ourselves with them and so on and so forth. But those that are not going to bother us, we'll probably leave them alone. The other group of organisms are the heterotrophic organisms, or the heterotrophs. And these, put it one way, is they require preformed organic compounds for their food. So they are dependent on those organisms that make those compounds. You and I and most of organisms that don't carry out photosynthesis, we are dependent. We're all dependent on preformed organic compounds, and hence we're heterotrophic organisms. Photoheterotrophs, they have the energy from sunlight, but the carbon is coming from organic compounds that are made by other organisms. Chemoheterotrophs, energy from carbon dioxide, energy and carbon from organic compounds. And they will process these molecules through respiration and fermentation. We'll talk about that later on. Now, I put the word hypotrophic or hypotroph there because we did talk about that in the previous lecture, where we talk about some organisms, not only are they heterotrophic, but they require living cells in which to grow. So maybe I consider them a subgroup of heterotrophic organisms. And then there is the saprobes, or the saprophytic organisms. These are also heterotrophic, but they derive their food source from dead, decaying matter. You'll find these organisms, they're able to produce some hydrolytic enzymes or lytic enzymes that will dissolve large macromolecules into very small ones, and they can bring that into their cell and use that as sources of energy. These organisms are really our friends. They are the ones that will help us with recycling of all those macromolecules that we cannot decompose by ourselves, but these saprobes can be considered huge decomposers. And so they like to recycle the matter, very essential for our living here on Earth. So I have a little review slide. I don't want to go too much without giving you a little bit of a chance to assimilate what we just talked about and see if you can understand. If you don't, go back, listen to the tape, look at the slides again. And of course, your textbook has a lot more that they, to offer than I can actually repeat here in the lecture. So please review that and we'll move on. Cultivation of organisms. Now we're going to cultivate. We know what type of, what they want, where they, how to group them nutritionally. And now we want to cultivate them. We want to really grow them in the lab. So before we start that, let's talk a little bit about some terminology. A culture. Culture usually refers to microbes that are growing on or in. So we'll talk about media in a second, because media is just food for the bacteria. So let's say we give the organism a broth medium, which is liquid. Okay. So Usually it's sterile broth, meaning no organism growing in there. It's just soup. So we will put organisms in there and see if they will grow. If they're growing, that growth of microorganisms we refer to as a culture. And when we talk about a pure culture, we're talking about only one type of organism, one species of organism in that particular culture. We will talk also about a mixed culture, and mixed culture usually have more than one organisms in the same broth or in the same medium, whatever that medium might be. A colony is a population of cells arising from a single colony. Now, we will explain this. You will see it in the lab a lot more than you will see it in the lecture. But just to give you a brief idea of what's going on here. If I inoculate it on a solid medium, on the surface of a solid medium, one single microorganism, that organism, if conditions are okay, will multiply over and over and over again. And eventually, it will form this huge big clump of microorganisms. Now I can see that clump with the naked eye. Remember, the one organism, which we measure in microns, we're not going to see with the naked eye. But when we have this big clump of organisms growing all in the same spot, millions and billions of them, then we see that big clump with the naked eye. That is what we refer to as a colony. We will learn later on that when organisms divide, the two of them are like duplicate copies of each other. So in a colony, an isolated or a single colony, you're going to have all the organisms there identical. And the importance of a colony will come in later on. Now, the microorganism itself that formed that clump of organisms is what we refer to as a colony forming unit. Okay, it's that one organism that formed the colony in the first place. Sterile, meaning no living microbes. I just mentioned the word before without explaining what it is. When we make media for the organism, which is the food for the bacteria, whether it's a broth or solid medium, we don't want any other organisms to be there. No organism there in the first place. So that medium is considered sterile, nothing growing on it. We are now going to put some organism on that medium and see if it will grow. That process of putting the organism in the medium or on the medium is what we call inoculation. We inoculate the organism on the plate or in the tube media. And we will use the tools of the lab in a micro lab, usually is an inoculating loop or inoculating needle. I'll show you how to use those in the lab, but we will use inoculating loops and inoculating needles to inoculate our media. Now, media is just food for the bacteria. We make media in the lab, whether it's we want to make solid medium or liquid medium or semi-solid medium, and we'll be using all of them. Bacteria, I don't think they care whether we feed them liquid or solid or semi-solid medium, as long as they get their nutrients. I don't think they care, but we want to identify, see how they're growing, what they look like, learn about them. And so we will make the media at different consistency. And one of the things that will help us to make the different consistencies, something we call agar. Now, agar is a complex polysaccharide, usually extracted from seaweeds. And it is used as a solidifying agent in our culture media, whether it's on Petri dishes, plates, slants, or deeps, we use the agar. And the reason why we're using the agar is because it's usually not metabolized by the organism. So the medium will remain solid and will grow on the surface of that solid medium or in between, depending on where we put it. Now, if we had made medium with gelatin, for example, gelatin can be used by the organism as a food source. And so before long, no gelatin is in the medium anymore and it becomes liquid. Now, suffice to say that agar works like gelatin, but it is not gelatin. It behaves like gelatin. If you boil agar, it will liquefy. If you cool it down, it will solidify. Just like you will do your jello. You want to make nice jello? You follow the package. You get your nice jiggly jello only after you put it in the refrigerator for it to set. If you leave it out at a very hot room temperature, it will still be liquid. So at certain temperature, the agar or the jello will be liquid. And if you reduce that temperature, it will solidify. And you can go back and forth. Heat them up again, they become liquid, cool them down, they become solid, and so on and so forth. So agar behaves like gelatin, but it is not gelatin. So then again, we use agar as our solidifying material. Now, media I consider the food for the bacteria. And like I told you before, we could have a liquid medium, a solid medium, or a semi-solid medium. It doesn't matter to the bacterium, I don't think. But I have a couple of words here: chemically defined media, where the exact chemical composition is known, and complex media is an extract made up of yeast digest, meats, partially hydrolyzed proteins we call peptones, or plant extracts and things like that. And you could have, we call them nutrient media. General culture medium is a nutrient broth or a nutrient agar. Broth, of course, is the liquid form. Agar, the solid form. And the agar, we can pour it in little dishes called Petri dishes and make agar plates out of them. Or we can put them in tubes and make deeps out of them. They solidify. Or we can even put the tubes in a slanting position and make slants out of them. We will be talking about a lot of media. That's why I don't give you too much of all the pictures and whatnot, because they're all in your lab manual, and we will be talking about them an awful lot too. Now, we can make media with added ingredients to it. And these added ingredients could make media selective. Some added ingredients will suppress the growth of some organisms and allow others to grow. In that sense, we're selecting what we want to grow and what we don't want to grow by using what we call the selective agents. Good examples are Macanchi agar, mannitol salt agar plates, and the PA agar plate, where we are having something in there, some added ingredients we call the selective agents that will prevent one group of organisms from growing, but not prevent all of them. So we're actually selecting what we need to grow, and we call that selective media. Differential media, on the other hand, we're also having some added ingredients in these media that will make it easy to differentiate one group of organisms from another, depending on some biochemical reactions. Again, we're putting into the medium what we call differential agents. And then we'll look for changes that occur in the medium that will help us, or will be the basis of differentiation, meaning it will help us to differentiate one group from another. In the lab, we talk about the Macanchi agar plate again, where some organisms will ferment the lactose and produce an acid that will be indicated by a change in a pH indicator. Other organisms that are growing on the plate may not ferment the lactose and hence will not produce acid, so will not change the color of the pH indicator. So we're going to have those that change the color. We're going to have a different color from those that don't. And that's where we can differentiate one group of organism from the other. In this case, lactose fermenting from the non-lactose fermenting organism. But again, I'm working in the lab with a lot of the media. So please, we're going to do that in the lab and it will be nice and clear, I hope, in the lab, because you'll be working with these media. You will be seeing results. You will be seeing the color differences and so on and so forth. And now I have another little review for you because I think we have done a couple of slides there for you to do another little review. Pause the lecture at this point, do your review, and then we'll move on. So now we want to talk a little bit about the environment. Remember what I said, for us to grow microorganisms, we need the proper food and then now the proper environment for growth. You can give the organism all the food they need, but if the environment isn't conducive to growth, they're not going to grow. And some of the things we look at when we talk about the environment for growth is the temperature, the pH, oxygen concentration, carbon dioxide concentration, and of course, osmosis, some barriers there, osmotic barriers. So we will go through each one of these in a little bit of details. But these are what we consider environment in which the organism will grow. Temperature, what we call cardinal temperatures. It's that range of temperature for the growth of a given species of microorganism. The minimum temperature, of course, as the name suggests, the lowest temperature that will permit the growth. Maximum temperature, the highest temperature that will permit the growth. Okay, and optimum temperature, of course, it is in between the two, but it is usually the most appropriate or the temperature at which the organism will grow the best. So that's what we call the cardinal temperature. Now, here is a grouping of organisms according to temperatures, and they have different groups according to temperature. The psychrophiles, the mesophiles, the thermophiles, hypothermophiles, and so on. And if you notice that there is a little bit of an overlap in the temperatures at which they will grow, but that's okay. So let's talk about each group. Psychrophiles, they grow optimally below 15 degrees Celsius. Remember, this is Celsius now, not Fahrenheit. So you should have an idea of what these degrees would feel like. They're capable of growth at 0 or even below 0. And when you store these organisms at refrigerator temperature, it's like they're very happy. They multiply very well at refrigerator temperature. You would think that refrigerator temperature will inhibit them, but no. Psychrophiles like that type of temperature. They grow very well. And I have some natural habitats there for you. You know, some of the lakes, the snowfields, the polar ice caps, the deep ocean, where it's very, very cold. They love that. Now, I put the word psychotrophs there, not to confuse you, but your book seems to want to mention that and talks a little bit more about psychotrophs than I would expect. But anyway, these are organisms that do prefer the cold, 15 below or below that, but they don't multiply at very, very low temperatures. If, for example, if you put these organisms in the refrigerator, you're not going to kill them, but then you might inhibit their growth. they will not be dead. But if you move them away from the refrigerator temperature and increase the temperature a little bit more to maybe instead of 4 degrees to 15 or maybe 12 to 15 degrees, they will grow very well. So they're not killed by the cold temperature, but they're not going to multiply at freezing temperature. So they're like, let's say the higher end of the psychrophilic stage, psychrophilic range, I should say. Now, psychroduric organisms are those, maybe they're regular organisms that like medium temperatures, but if you put them in the cold, you're not killing them. They can endure, they can withstand a little bit of that cold. Okay. Now, the mesophiles, those are the ones that like moderate temperature. They grow between 20 and 40 degrees. And usually, many of our human pathogens are in this group because your body temperature, you all know, is about 37 degrees or so, smack in the middle there. And so if they can withstand your body temperature, they can be potential pathogens. So a lot of the organisms we are going to be dealing with in our labs are going to be in this particular category, the mesophiles. When we do the effect of heat on microorganisms in one of the labs, we will be cultivating some of the thermophiles and the psychrophiles. But in general, because these are the ones that will be causing human diseases, we will work with them a lot more than the others. Now, thermophile, the names will tell you what they are. Phile meaning like, thermo, heat. And these organisms grow optimally, meaning best at over 45. And they will, I give you some natural habitats there. A lot of times you find them in compost piles. Now, I don't know if you know it, but when you're doing compost, sometimes that temperature, once the organisms are working to decompose the larger molecules, the larger macromolecules, temperature could get very, very high, can be very hot in there. Okay. And most of the bacteria that will be pathogenic to us will not be succumb to organisms that are thermophiles because they don't like our temperature. They don't grow at our body temperature. Thermophiles like a lot more heat than that. You'll find them maybe in volcanic craters. Where else could you find them? Naturally, you'll find them in hot springs, for example. If you see, the geysers, the water that's spewing out of those geysers, sometimes you can actually see steam coming out of them. They're really not boiling. I don't think the water is boiling, but at least very close to it. These organisms will survive very well in those hot temperatures. Extreme thermophiles, the name will suggest that they like extreme heat, very, very high temperatures. 100 degrees Celsius is boiling. Going to 121, that's like putting something in, very hot in, where would be in our homes, maybe a huge pressure cooker or autoclaves in clinical settings can be set to 121. That's very, very high temperature. All right, thermoduric, again, like psychroduric, they don't like the hot temperature, but they can survive short exposures to hot temperature. Some of the thermoduric organisms can survive when we do pasteurization, when we pasteurize our milks and our juices, we heat them up to about 63 degrees Celsius for a half an hour. That's as one step of, or one way to pasteurize stuff. Another way is to heat them up maybe to 71 degrees for about 15 seconds or so. But even that high temperature is enough, it's not enough to kill some of the organisms that are resistant to that temperature. So These are the thermoduric organisms that can withstand the high temperature, but are not killed by them. But they're not thermophiles. They don't like the heat, but they can withstand the heat. Before I go any further, I think I have a little animation here for you. So why don't we look at it and see what it's all about?
Microorganisms reproduce by fission. Each parent cell increases in size and then divides to form two daughter cells. The progression is swift. 2 become 4, 4 become 8, 8 becomes 16, 16 become 32, 32 becomes 64, and so on. The growth of microorganisms depends on conditions in the environment. The main factors are temperature, moisture, nutrients, pH, oxygen, and chemical inhibitors. Each microorganism has very specific needs in each of these categories. The growth of microorganisms follows an exponential pattern. If there is sufficient water and nutrients and a warm enough temperature in the microorganism's environment, and the other three conditions are right, multiplication is extremely rapid. Under ideal conditions, the generation time for multiplication of bacteria can be as short as 30 minutes. So, in 24 hours, in the right environment, one bacterium can produce over 1 billion offspring. Of all the environmental factors that influence bacterial growth, environmental temperature is the most important. The following graphs depict the change in growth rates of typical microorganisms as a function of temperature. Here is a typical graph showing the increase in numbers of cells of the bacteria called E. coli as a function of time at the optimum growth temperature for the organism. Now watch the way that higher temperature affects the survival of E. coli. As the temperature is increased above the maximum growth temperature, the E. coli cells begin to die. As the temperature is increased, the cells die more and more rapidly until all cells are dead at temperatures above 80 degrees Celsius. Now watch the way that lower temperatures affect the growth of E. coli. As the temperature is decreased, the rate of growth slows until at some low temperature, slightly below 6 degrees Celsius, the cells cease to divide. The cells are still alive and waiting for the temperature to increase so they can begin to divide again. It is the cessation of growth of microorganisms at low temperature and their physical death at high temperature that defines the safe temperature ranges for the storage of foods.
All right, so that's a little bit about the effect of temperature on the growth of microorganisms. Now, let's talk a little bit about pH requirements. And we can group microorganisms depending on pH requirements as acidophiles, alkalinophiles, or mesophiles. Or, I'm sorry, neutrophiles. So acidophiles, the name suggests, philes liking, acidophiles, they like acid. Organisms that thrive in acidic conditions. Many of the fungus, the yeast and the mold, they tolerate some acidity. Now, they don't like very highly acidic conditions. Yeast, molds, when we make media, we adjust the pH to about 6.4, which is not very highly acidic. But some of them can actually grow in highly acidic conditions, the acidophiles, and they are the ones responsible for spoilage of acidic foods, like your pickled foods. Okay, your slide here shows about a couple of organisms that can grow best at pH 1 to 2. That's very, very highly acidic. Alkalinophiles, on the other hand, they thrive in alkaline conditions. And here you have some Natromonas that could live in soils at pH 12, very highly alkalinic conditions. And there are some like Proteus that maybe they don't find the alkalinic condition, but they can make it for themselves. Proteus, actually, we'll talk about it a little bit more in the lab, where this organism is a pathogen of the urinary tract in humans. Now, human urine is actually slightly acidic. Proteus don't like the acidic condition. So what it does, it's able to create or to make an enzyme called urease. And it actually breaks down the urea in the urine to form ammonia. And ammonia will make its surroundings alkalinic. So the organism can grow very well there. Majority of the organisms we're going to be cultivating in the lab or learn about, they grow between 6 and 8. Most of the time, we call them the neutrophiles. I don't have a slide for the neutrophiles, but if we go back, like I said, the majority of the organisms, they like pH between 6 and 8. Those are the ones we call neutrophiles. Maybe I can put the word there for you, so you know what we're talking about. And most of the organisms that are going to be pathogenic to humans will also fall within this category. So we'll be learning a lot about them. In fact, when we make media in the lab, normally we will adjust the pH of the medium to 7 so that we can cultivate most of the organisms we want to learn about, the neutrophiles, and see how they react to the different media. Osmotic pressure, osmophiles, they live in habitats that are high in solute concentration. Let me give, sometimes it's salt, sometimes it's sugar. In fact, halophiles, they like a lot of salt. They prefer very high salt concentration. Halobacterium, halococcus, they could survive in solutions that are 25% concentration of sodium chloride. We will talk a little bit about Staph aureus in our lab. Well, not just Staph aureus, but all staph species in the lab. We will cultivate them on what we call mannitol salt agar plate. And they grow very well on the mannitol salt, which is about 7.5% sodium chloride. So if we increase the amount of salt in a medium, we can the more we increase the salt to a point, we can actually prevent other organisms from growing and allow only the ones that are halophiles, the ones that like the salt, to grow. In fact, aren't we just preparing some now selective media? Just by increasing the salt, we're preventing all the others from growing, except only those that like the salt will grow. So we have actually created a selective medium using salt as one of the ingredients, the selective agents. And like I said, also, you can have sugar. You know that a lot of people use sugar to preserve. Many times when you don't have any other way to do it, you can preserve things in very like syrupy stuff. Cherries, for example, you can dump in a lot of very high sugar concentration and you can preserve them for a very long time. I think I went backwards. All right. So now we're going to go to another requirement, environmental requirement, and that's oxygen concentration. And we can group microorganisms according to their usage of oxygen as aerobic, anaerobic, facultative anaerobic, microaerophilic organisms. And we're going to go through each one of those a little bit more in details. Aerobic organisms, they use oxygen, gaseous oxygen in metabolism. And usually when you do use the oxygen as the final acceptor of the electron, In that process, not only do you produce water, which is non-toxic, but you can produce some toxic oxygen products, like some of the superoxides. And you need enzymes to process or to detoxify those toxic products. So aerobic organisms, they use the oxygen and it's good for them, but they can also produce the enzymes to detoxify the superoxides. Anaerobic organisms, they do not use oxygen as their final acceptor, but some other organic or even inorganic material, but not oxygen. And one of the reasons is because they lack the enzyme for processing those toxic oxygen byproducts. And if you don't have the enzyme to process those, then you're going to get killed because of the toxicity of those toxic byproducts. So obligate anaerobic organisms, not only don't they use the oxygen, they don't have the enzyme, they lack the enzyme to process toxic oxygen, and they will die in the process. Aerotolerant anaerobic organisms, they're not harmed by the oxygen. They don't use the oxygen in metabolism, but they're not harmed because they can find alternate ways of breaking down the peroxides and the superoxides that are produced. So they're not really harmed by them, although they don't use the oxygen. I mean, the name is telling you, aerotolerant, they can tolerate. When we say aero, we're really talking about the oxygen in the air. So they can tolerate the oxygen. Facultative anaerobic organisms, they may or may not require oxygen. If oxygen is there, they'll use it. If oxygen is not there, they'll find other ways to go through their metabolism. But if oxygen is present, they prefer that they grow better. And we'll learn later on in the next lecture that when oxygen is present and they could go through respiration, they can produce a lot more energy than when oxygen is not present and they find alternate ways of metabolizing their food products. Microaerophilic organisms, they don't want, microaerophilic is telling you, they like microaero, small amounts of oxygen in the air. Now, we know that the concentration of oxygen in the air is about 21%. They don't like 21%. They like maybe about 10% or so. And that's why we call them microaerophilic. Capnaic organisms, on the other hand, they are microaerophilic in a sense that they not only like small amounts of oxygen, but they prefer a large amount of carbon dioxide. Now, the next couple of slides, we'll talk a little bit about how we create these conditions for these groups of organisms to grow. For example, anaerobic organisms, they don't want oxygen in the air. They're not going to grow when oxygen is present. And so we have to create an environment that is free of atmospheric oxygen. And how do we do that? Well, one of the ways to do it, and there are many other ways to do it, but a couple of ways I'm going to talk about it right now, is to put our plates that have been inoculated with the organism, anaerobic organism, and seal the jar. Okay, we seal the jar, but we also include in that jar a couple of things. A packet, which is a gas pack, they call it, a CO2 and H2 generator. And you also have some catalyst pellets there. Remember, once you seal the jar, you're sealing some air in there, but no other air is going to go in, sealed. So the air in the jar already has 21% oxygen. We don't want the oxygen in there, so we have to remove the oxygen. And we do that by combining the oxygen in the air in the jar with hydrogen. That will form water because we have a catalyst there to help us out with that reaction. The water will condense to the bottom of the tubes, the bottom of the jar and the plates. But the point is, we now have an oxygen-free atmosphere and our anaerobic organisms can grow. Another way we can cultivate anaerobic organisms is to use something called a reducing medium. And we have a couple of reducing media that we use in the lab. Thioglycolate, for example, is a good one that we normally will use in the lab to cultivate our anaerobes. Now, what's going on with this medium? And thioglycolate is a liquid medium. And you know, in liquids, a lot of gases, including oxygen, can dissolve. So if the oxygen is in the liquid, obviously we're not going to be able to cultivate anaerobic organisms. But if we put in this medium a reducing agent, and remember we talk about that in one of our lectures, I think the second lecture, we talk about reducing agent. Reduction means one of the definition is to take away oxygen. So the reducing agent is in the medium to take away the oxygen. So we're putting that chemical in there. As soon as the oxygen dissolves in the medium, in the liquid, the reducing agent would absorb it and get it out of the way. And if it can do that, then now we're freeing, the medium becomes free of oxygen, so anaerobes can grow. And that's another way by using a reducing medium. Now, carbon dioxide, I told you just now that there are some organisms we group as capnophiles. They like a lot of carbon dioxide in the atmosphere. Now, there's only about 0.03% carbon dioxide in the atmosphere. These organisms like 3 to 5%, hundreds of times more than what's in the atmosphere. So how do we do that? Well, one of the ways we do it is to create conditions that is, well, capnaic. The first one is capnaic and microaerophilic at the same time. And how do we do that? Here we have a jar again, and we seal it. It has a lid that we can seal. And we can put our plates that have been inoculated with our organisms and our tubes. But before we close the jar, we put a lighted candle in there. Now, what's going to happen is, like, remember, the jar is sealed. So no more air is going to go in. But the candle is going to burn. And what's going to happen is that as the flame, the combustion is going to use up the oxygen in the jar. And at the same time, will emit carbon dioxide. So we're creating more carbon dioxide and at the same time reducing the amount of oxygen. Perfect conditions for some of our capnaic organisms to grow. And some students will argue that, well, wait a minute, we have the candle in there. Wouldn't the candle burn out all the oxygen? No, that's not true. If that's the case, we can grow anaerobes in there and we won't be able to do that because that candle will go out long before we use up all the oxygen in the jar. Because combustion needs a certain percentage of oxygen to take place. Otherwise, it will not. Okay? So I sometimes relate it to a burning building, for example. You see on TV many times. When you see smoke coming out of a building, you call the fire department. And it's all smoke at that point. It's smoldering, but you don't see the flame. The minute the fire department comes and the guys go there and they break the windows and the doors open, then you see the flame because now there is oxygen to feed that flame. Before time, it was smothering because there's not much oxygen in there. Okay? So no, you're not using up all the oxygen when you put a lighted candle in there. The candle will go out before you use up all the oxygen. In the second picture there, it's like a little package. And in this package, we have a CO2 generator. So it will increase the amount of CO2 in the package. And so we can create some capnaic conditions to grow capnophilic organisms. Okay, so these are some conditions for growth. Again, another bit of a review slide. You can pause the lecture at this point and we can continue when you have done your little review there. I just want to make sure that you understand what the slides are all about. So I don't go through too many slides and just give you one review in the end. At every point, so many slides, I try to give you a little bit. Now, reproduction of microorganisms. I told you that microorganisms will grow. And if we're dealing with bacteria, and in fact, the rest of the slide, we're really talking about bacteria, the prokaryotic cell. When these organisms grow and they grow in binary fission, one of the animations I gave you earlier in the lecture talks a little bit about the binary fission process. You saw that. We'll go through that again, but it will go through binary fission and multiply over and over again until you see a colony of bacteria. Okay, so when we talk about reproduction of bacteria, we're not talking about the organism getting bigger and bigger, but we're talking about the number of organisms getting bigger and bigger. And reproduction in bacteria, remember our prokaryotic representative is the bacteria. We can have binary fission, budding, conidiospore production, and fragmentation of the filaments. I think the very, very first lecture, we had a little animation on that shows a little bit of budding, conidiophore, and fragmentation. But this time, we're basically going to concentrate on the binary fission process. And like I said, I showed you a little animation before. I'm sorry. I have another one here. So maybe you look at the slide and it says 1 cell becomes 2 cells and 2 cells become 4 cells and so on and so forth. And you're actually going through the process. And every time 1 cell becomes 2 cells, a little bit is happening. The parent cell will enlarge, chromosomes are duplicated, and then the cells will start to pull apart until you eventually get 2 complete cells being produced. For me to explain all that to you, I think an animation will be a lot better. So if I can get your attention here for a moment, let's see what's going on here.
Bacterial cells generally increase in numbers by the process of binary fission. The cell increases in length while doubling all of its parts, then forms a cross septum and divides to become 2 cells. 2 cells become 4. Those 4 become 8, and so on. This pattern of increase results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in numbers every 30 minutes. Other bacteria have generation times of several hours or even days. During growth of the cell, streptococci and some other gram-positive cocci synthesize new cell walls in the septal region. Rod-shaped bacteria such as Escherichia coli, Salmonella, and Bacillus species synthesize new cell walls over the entire surface of the cell.
So that gives you a little idea of what binary fission is all about. I have a couple of slides there that will show you the same thing, but I prefer the little animation. They're generally more explicit than I can be. So all these words, generation time or doubling time, those were explained in the little animation. You can go back to that slide and look at it again if you want to, but they're all there. Most of the organisms we use in the lab will have a short generation time because, you know, we want to have nice colonies by the next time we come to the lab when we try to cultivate microorganisms. For example, your E. coli and your bacillus and your staph, generally 15, 20 minutes generation time. And so by the time we come back the next day, we have billions of microorganisms. Now, this slide talks a little bit about the growth curve of bacteria. And we're talking about this number here, the red line, represent the number of viable organisms, live ones, that we cultivate. Now, for you to understand what's going on in this graph here, we can picture a tube of media, let's say a tube of broth media, that we have inoculated some organism in. And let's follow what goes on with that tube of media and the organism you just inoculated. Let's say we inoculated E. coli in this tube. What happens? Now, here's the time, as the time improves or the time increases, the number of organisms, the number of live bacteria will increase too, to a point, and then you notice it will go back down. The reason for this happening is because now we have a finite amount of media, meaning a finite amount of food for the organism in a finite compartment, in a tube. And then we're going to follow the growth. So let's say we inoculated a certain amount of organism in the tube. And we're going to end up with what we call a lag phase, and then an exponential or log phase of growth, then a stationary phase of growth, and then the death phase of growth. Let's see what's going on. And I think I have a little explanation for each of the phases. Now, in the lag phase, which is that flat phase at first, remember, you put in a certain amount of organisms there. So you're going to have that amount of organisms still there. But the organisms are really not growing yet. They're not multiplying because the number remains the same, kind of. It's flat. Why is that so? Well, in every situation, it makes sense. If you go into a new neighborhood or the organism is in a new surrounding now, it's not just going to multiply beginning right away. Usually they want to make sure they scout around the surroundings, they look around, they make sure it's safe, everything is okay, environmental conditions are fine, the amount of food is fine, the type of food is okay. And so it takes a little while for them to get used to their environment then. And that's why we call it the lag phase. They're not multiplying yet. But once they realize that, hey, temperature is right, pH is right, food is right, environmental conditions right, then it's okay. They will start to multiply. And they will go through this binary fission every generation time. One organism will split and give me two. So the numbers keep on multiplying. Keep on increasing and increasing logarithmically. Not 1, 2, 4, 6, 8, and 10. No, it's 1, 2, 4, 8, 16, 32, 64, so on and so forth, logarithmically. And then you're going to have, within a short period of time, a lot of microorganisms. It hits the point where it seems like they stop. The number of viable organisms remain the same. It becomes flat again. So this is the log phase, flat portion where they're getting used to. And then you have the logarithmic or exponential growth phase. They're increasing geometrically. And then all of a sudden you hit that stationary phase, which is the flat stage again. They call it the stationary phase because the number of viable organisms remain the same. Why? Well, it seems like, remember, you still have this limited amount of medium in a tube space. So what's going on? When you have a lot of space, you have a lot of food, the right environment, organisms will continue to multiply. But it gets to the point where now so many organisms in the tube, population density is increasing. It's the same amount of food. So if you have so many times more microorganisms there, and the same food supply that everybody's depending on, then food becomes, after a while, very scarce. Okay? And sometimes, some organisms, the survival for the fittest, they will run for the food, they will try to get the food. And the others who are not as aggressive might just fall behind and die of starvation. Not only that, not only are they dying of starvation, remember, you're going to have an accumulation of waste products in that tube too. So a lot of things, environmental conditions are not favorable anymore. So it's a flat number there. It's not rising or falling, it's flat, because it seems like the number of organisms that still can find food, a little bit of food that's there, and multiply, will balance the number of organisms that can find the food and die. So you have the number of viable organisms remain the same. But as conditions become more and more unfavorable, less and less food, more and more toxic waste, more and more live dead organisms, no space there, less space for each organism, you end up with what we call the death phase of growth. Okay? And the death phase, if you look at it, seems very similar to, or opposite, I should say, to the logarithmic phase, where the organisms are dying off at a rapid rate. Okay, organisms are dying off because lack of food, accumulation of waste, no space. And so, yes, organisms are going to start. Conditions are becoming very, very unfavorable, and organisms will die. Notice that not all of them die. It just turns out that some organisms in the death phase, some organisms that can produce some survival mechanisms, will try to evade this very unfavorable conditions, maybe by producing spores or something like that. And then when things improve again, they will start to multiply. All right? But that is basically what we call our growth curve. A phase where they get used to their environment, things are very okay with them, they start multiplying out of proportion, and then finally realize, well, wait a minute, we don't have an unlimited supply. We only have a certain amount of food, a certain amount of space. And so some organisms will still fight and find that space and food, and they will multiply, others will just die. But eventually, a lot of the organisms will just die off because no food and no supply of no food, no space, and just an accumulation of waste. And they will probably all, not all, but most of them will just die, except if you have the ability to form spores and things like that. All right. So that's where I have the growth curve. I think that is the death phase. And then we're going to do a couple of slides more there to measure the growth. How do we measure growth? Well, a couple of things we can do. If we see or we inoculate a liquid medium, usually the liquid medium is very clear. And once the organisms start to grow, it is going to be very cloudy. So turbidity or cloudiness is an indication of growth. So very crudely, we can say the more turbid the medium, the more organisms are there. Because remember, the organisms aren't getting bigger, they're getting larger in number. So more turbidity, more organisms. Okay? And so we can measure using a spectrophotometer how turbid the medium is and figure out what number of organisms might be in the tube. And that's a very crude, it's not accurate, but a very crude estimation. And we can do the counting. We can directly count the number of organisms. I mean, one of the things we do in the lab a lot of times when a doctor sends for, you know, a patient blood sample for a CBC, we're counting the number of red cells in a milliliter of blood. And you know, in a normal human, body, you have about 4 to 5 millions of red cells in that 1 ml of blood. And we're expecting to count that in the lab. But there is a way to do it. There is a way to do it. So we can do either a direct count. We can put some organisms under the microscope, a certain volume, and we can do that and actually manually count and adjust for the volume. Or we can use something called a Coulter counter, which will electronically, it's almost like a little computer that will electronically scan a fluid for these tiny particles that are in there. I'm not going to go through how that works, but we can do that for red cells too. We can count using a Coulter counter, or flow cytometry will also help you to do some direct counting. You can do some filtration, but before the filtration, maybe I should show you how we can count directly. We have something called a Coulter counter there, a chamber, and it can only hold a certain value. And you can actually fill this up, carefully fill it up, that certain volume of broth in there, and look under the microscope and directly count the number of organisms. And then, depending on the number you have, you adjust for the value. Okay? You can estimate the number of microorganisms. Well, actually, you're counting the number of organisms that are there. Another way to do it, like I said, was using the spectrophotometry method, where if you have turbidity in the broth medium, less light will go through. More light will be absorbed. And so depending on the transmitted light or the absorbance, you can actually figure out how turbid this medium is and actually directly measure the amount of organisms in there. And I think the last slide tells us a little bit about the filtration method, because microorganisms are tiny, yes, but we can have filter paper that will read. The pores are not that large. They're very, very tiny, and microorganisms will not be able to go through. When I say microorganisms, I'm talking bacteria. We'll learn later on that viruses can go through because they're really very small. But bacteria will stay on the filter paper, and then we can cultivate what's on the filter paper. And actually, remember, every colony there is almost like one organism that developed into a colony. Remember, an organism might be looked upon as a colony forming unit. So we can actually cultivate the organisms, count the number of colonies, and figure out how many organisms were there in the first place. I thought that was the last slide, but there was another one, indirect methods to estimate number of bacteria. In fact, estimating growth, metabolic activity, or dry weight. What do I mean by metabolic activity? Well, we will learn later on that there are many byproducts that are produced when organisms go through certain metabolic processes. For example, some organisms may ferment a sugar and produce acid. If we know that one organism produces a certain finite amount of acid, we're probably not going to use one organism, it's too tiny to work with, but we can use a colony of organisms. And depending on the amount of acid produced, we can estimate how many organisms are there to produce that amount. Same thing with the weight. We're not going to be able to weigh one organism. No. I mean, not in a clinical lab. You can do all these things if you have those sophisticated machines these days. Of course, that's all possible. But in a regular lab, we're not going to have that. So if we know what one organism weighs, and we have tons of organisms that we can dehydrate and weigh them, then we can estimate from that amount of organisms we have, I mean, the weight, how many organisms are in that container. So those are some indirect ways of measuring growth. All right. And I think my last slide, I hope this is the last one, is a little bit of a review for this part of the lecture. So please, again, go through the slides, go through the review, all the review slides and see what's there. If you still don't understand anything and you go through the lecture again or your book and you still don't understand, please bring those questions in when you have our office hours so we can discuss them and, and try to clarify whatever is bothering you. All right. So please, I appreciate your listening, and we will stop the lecture at this point and we will continue to another lecture very soon.